Unit 2 Review, Honors Biology. You can find this review and this video at www.srhsbio.wikispaces.com.
First question asks us to compare and contrast two enzymes used in the body. In the response, explain the importance of each enzyme, describe what will happen to the organism if it lacks or has a deficiency of each enzyme, and agree or disagree with the statement, enzymes are essential for the health of an organism, and explain why. So first enzyme that we're going to talk about is catalase. Catalase catalyzes the breakdown of H2O2, hydrogen peroxide, which is a toxic waste product made by the cell, into harmless water and oxygen. This is found in all cells that go through cellular respiration, aka breathe oxygen. So if this is my catalase enzyme, it's going to fit with hydrogen peroxide. Whenever it does that, it breaks down the hydrogen peroxide into water and oxygen. Next enzyme is salivary amylase. This breaks down polysaccharides into disaccharides, beginning the process of digestion in your mouth. It's produced by your saliva glands. So salivary amylase fits perfectly with polysaccharides, with starches. Its job is to take those starches, those polysaccharides, and break them apart into disaccharides. So our starch, our four sugar starch, breaks down into 2 disaccharides. Another type of enzyme is pepsin. Pepsin begins the digestion process of proteins in the stomach. It takes proteins and it breaks them down into smaller peptide chains. And it does this by only cutting between a certain pair of amino acids. There's different enzymes that cut between different pairs of amino acids. This just makes the proteins a little bit more sizable. That way they can be absorbed easier during digestion through the walls of the intestinal tract. So pepsin fits with our polypeptide chain there. Each of those little different symbols represents an amino acid. Causes that chain to be broken apart into 2 smaller protein chains. So here we see we've cleaved between a specific set of amino acids, in this case the heart and the green star. Trypsin is another enzyme that breaks down protein, except this one cleaves between a different set of amino acids. This is produced in the pancreas to aid in protein digestion. So there again is our polypeptide chain. We see that it fits onto a different pair of amino acids. And in this case, it's going to cleave right between those 2. So in this case, I got a amino acid and a smaller protein chain. This just makes these little protein pieces a little bit smaller and a little bit easier to be absorbed by your intestine during digestion. Last one is pancreatic lipase. This breaks down lipids into glycerol and fatty acids, again allowing them to be absorbed by the intestine. It's made in the pancreas and secreted into the small intestine specifically to break up lipids. So it fits perfectly with a lipid. So there we got glycerol and 3 fatty acids. And it's going to take that and break those up into their pieces. So now these much smaller pieces are easier to be absorbed by the stomach and by your digestive system. So in summary, catalase works on hydrogen peroxide to produce water and oxygen. It is produced in all cells, and its importance is that it helps prevent a toxic buildup in cells by breaking down that peroxide. Salivary amylase, that is an enzyme that acts on polysaccharides or starches to break them down into smaller disaccharides, which means 2 sugars. This is produced in the saliva glands in your mouth, and it begins digestion of starches in the mouth. That means that if you eat something like crackers or something that's made of carbohydrates, the amylase in your mouth will break that down. If you were to eat something with protein in it, there's nothing in your mouth to break apart protein, so that would just sit there and rot until the bacteria got to it. So it's a good thing that we have salivary amylase to help speed up digestion. Pepsin digests proteins, polysaccharides, breaking them down into smaller polypeptides, smaller chains, and amino acids. This is found in the digestive system, and it helps cut proteins to make them easier to absorb during digestion. Trypsin has similar properties. It also breaks apart proteins. It just cleaves between a specific set of amino acids. It's also found in the digestive system, and again, makes those proteins a little smaller and easier to be absorbed by your digestive tract. Last is pancreatic lipase. This works on lipids to break them apart to fatty acids and glycerol. It is produced in the pancreas, and it helps to cut lipids up so that they can be absorbed during digestion. Some similarities between all of these. Most enzymes are going to end with ACE. So here we see that these end with ACE. And a lot of times the name can help you indicate what it does. So lipase probably breaks down lipids. So check for, whenever you see names, check for if it ends with an ACE. A lot of times the problem will tell you if a particular substance is an enzyme or not. Next question asks us to consider a diagram and respond to the questions that follow. So the diagram pictures a cell, and that cell is surrounded by a bunch of different chemicals. A is water, B is salt, and C is some large substance that is needed by the cell. And there's a little note on here, assume that the cell membrane has a protein to allow A in and out. So we got a few questions to answer here. So the first thing we're going to do, we're going to take a look at this cell, and we're going to go ahead and zoom in on the cell until all we see is the cell membrane. The cell membrane is made up of phospholipids. Phospholipids have a phosphate head, which is a hydrophilic, water-loving region. And then they have two fatty acid tails. These are hydrophobic, which means water-hating. So now that we got those basics down, why can't salt pass through the cell membrane? So there's salt. It's made up of sodium chloride. This is an ionic compound, which means that this side is positive and this side is negative. This is a small charged particle. So here's my salt. And if my salt tries to get into this cell membrane, it's going to be repelled by those fatty acid tails. So the hydrophobic parts of the cell repel charged particles, such as water and salt. So salt can't pass through the membrane because it's a charged particle. Next question, if the salt can't move into or out of the cell, how will the water move? So here's the picture of the cell that you guys have in front of you. I'm just going to simplify it, and I'm going to only look at water and salt. So I'm going to take out all of the C's. So if I go through and I count, I have 5 salt particles inside, 1, 2, 3, 4, 5, and that's all of the particles. On the outside, I've got 1, 2 salt particles, but a total of 10 other particles. So if I were to take these numbers here and make them into a ratio, I would get 5 over 5, which is equal to 1, and I would get 2 over 10, which is equal to 0.2. So we can clearly see that the amount of salt inside and outside of the cell is not even. So what's going to happen, because there's a higher concentration of salt inside than outside, water is going to flow into the cell. And it's going to do this until the concentration of salt inside and out of the cell is the same. So it'll look something like this. to prove to you that it is the same concentration inside and out. If we count the salt particles inside, we get 5. There's 1, 2, 3, 4, 5. The total number of particles inside the cell is 10. So if we do 5 over 10, we get half, 0.5. On the outside of the cell, there's 2 salt particles, but a total of 4. So 2 over 4 gives us the same thing, 0.5. So even though there's more salt inside of the cell, there's also more water. And that balances out so that the concentration of salt inside and outside of the cell is the same. So since water is doing the moving, let's explain that process. Is energy needed for this process? So there's my cell again. I'm going to go ahead and zoom in so we see that cell membrane. Osmosis is the facilitated diffusion of water across a semi-permeable membrane. So there's my water. There's a bunch of it trying to get into the cell. So you see it bounces around in there, and it, by random diffusion, by random motion, can bounce through that aquaporin and enter the cell. Here's a short video to show that. This is the cross-section of a cell membrane, so that's similar to the picture that I have here. There we see the hydrophilic heads and the hydrophobic tails. So in order for something to get through, it's going to need an aquapore to allow water to pass through the membrane. So this blue thing in the middle, that represents our aquapore. It's made up of 4 subunits, and each subunit has a little channel that water can go through. We're going to go ahead and zoom in on just one. So we can look at the surface of the protein, and if you look at that, you see there's this tiny little hole in the middle. It's a very narrow channel, but water molecules are very tiny, so they fit through quite easily. This is a protein. Here you see that the little blue balls there, that represents water moving through the channel. So that's what an aquapore is. Next question, why can't substance C just diffuse into the cell? So once again, we can go ahead and zoom in on our cell membrane. Substance C is a large substance needed by the cell. It's too large to diffuse through the cell membrane. So even though it's not strongly charged, it's just too big to fit through the membrane. So if it tries to come in contact with the membrane to try to get through, it's just going to bounce off because it's too big. Explain how substance C will get into the cell. Is energy required for this process? So active transport is the process of moving molecules into or out of the cell by expending cellular energy, which is called ATP. There's 3 types of active transport that we focused on. There's endocytosis, exocytosis, and protein pump. So first I've got a short video that shows you in very simple terms what endo and exocytosis are. So here I've got a string that represents the cell membrane. The tennis ball is something that I want to get inside. So the cell membrane can engulf the particle that it wants, in this case the tennis ball. That's endocytosis. And once it's done, if this was food and it breaks it down into waste, it can expel it. In that case, you see the softball being expelled. The next type of active transport is a protein pump.
In this case, the cell uses transport proteins which need a boost from an energy molecule. This actually changes the shape of the proteins, causing them to pump different molecules and ions across the membrane. Now from the side with fewer solutes to the side with more.
So in that little video, the green structure was the protein, and you saw that every time it touched with one of those little pink lights, it changed shape. Those pink lights represent cellular energy, or ATP. So to get substance C, that big substance, that's going to be like the picture where we saw the tennis ball being taken in, which is endocytosis. Cyto is a prefix that means cell. Endo means into. Exo means out of. So endocytosis into the cell, exocytosis out of the cell. And of course, protein pump is pretty straightforward as to what that means. So consider the experiment below. Write a statement about what pH catalase is normally found in based on the information in the picture. So here I've got my pitcher, and this is filled with some hydrogen peroxide, and it's at different pHs. I've got neutral, strongly acidic, strongly basic, weakly acidic, and weakly basic. And inside is a little piece of liver, which contains the enzyme catalase. Catalase breaks down hydrogen peroxide into water and oxygen. So we see the bubbles in the tubes, and that is the oxygen gas being named. The tube with the most bubbles is breaking down the most hydrogen peroxide. Therefore, catalase must normally be found in pH 7, which is the neutral solution, because it functions best at this pH level, evident by the high number of bubbles that we see. Draw a graph that shows 3 enzymes, one that functions best at pH 2, pH 7, and pH 10. So, what we'll see, we've got along this axis here, I've got pH, and I've got enzyme activity along here. So, an enzyme that works best at pH 2 is going to show the most enzyme activity right around pH 2, and then it will taper off. So that's right around pH 2. Go ahead and do the same thing, this time for pH 7. So this is going to show the most enzyme activity around pH 7. And now we're looking for one that functions best in a basic pH around 10. So again, I'm looking to draw a line that shows the most enzyme activity, this time right around 10. So that's 10, and this one is 7. So, which enzyme best represents the functions of present in most mammals? So, in most mammals, it's going to be right around pH 7. And again, this depends on what the enzyme is. Some enzymes are found in your stomach, which is more acidic, obviously, but generally speaking, pH 7 is what we expect to find in most mammals. So, how could I slow down the rate of enzyme activity for the pH 7 line? So we see that this works best at pH 7, which means that if I change the pH to something else, either to make it more acidic or more basic, that's going to decrease the functionality of this enzyme. So if we were to change, say, the pH to pH 3, okay, here it's got very low enzyme activity as compared to what it's got at pH 7. How does temperature and pH affect the shape and functionality of an enzyme? So each enzyme has a unique set of conditions under which it will work the best. Altering those conditions can alter the shape of an enzyme, decreasing its activity. So here I've got salivary amylase, and that works best right around body temperature and a pH of 7 because those are the conditions that we find in your mouth where salivary amylase is made. If I were to change those conditions, I would cause the enzyme to be denatured, and you see that the shape no longer fits. So with the regular salivary amylase, we can see easily that it will fit with this starch. If it becomes denatured by changing the temperature, it's going to mess up the shape, so it no longer will fit with starch. So if I take that amylase and I boil the enzyme, or I put it into a basic pH, I'm going to denature it, preventing it from fitting with the starch and breaking it down. So how does an enzyme speed up a rate of reaction and then draw two lines to represent the rate of reaction with and without an enzyme? So enzymes are catalysts. They're biological catalysts, and they speed up reactions the same way any catalyst does, by lowering the activation energy needed for the reaction. This is shown on the graph by a smaller hill. So the blue line represents the reaction without an enzyme. It's got a higher hill. The red line represents the reaction with an enzyme. You see the hill is lower. That shows the magnitude of the activation energy without the enzyme and the activation energy with the enzyme. So you see that the blue line, the hill goes up almost twice as high as the red line. So that one needs much more activation energy, therefore it's going to be much slower. These both represent the same reaction, just one is without an enzyme and one is with an enzyme. So the macromolecules. First one is carbohydrates. The monomer for carbohydrates are going to be monosaccharides, or simple sugars. When you get a bunch of those together, you get a polysaccharide, known as a complex sugar or a starch. This is primarily used as an energy source, and there are some structural purposes. So glucose is blood sugar. That's your body's primary source of energy. Structural purposes include chitin, which is what exoskeletons are made of, like in horseshoe crabs or grasshoppers. Cellulose is another structural carbohydrate, and it's used for plant cell walls. The monomer for phospholipids is going to be fatty acids. It's also made up of a phosphate group and a glycerol. The polymer is a phospholipid chain, so I string a bunch of those phospholipids together, and I get a chain of phospholipids. If I put two of those together, a phospholipid bilayer, bi means two, I get a cell membrane. So that's what's around, say, your heart cells or your skin cells, or it's the membrane around a single-celled organism. Those are all cell membranes. The monomer for fats or oils is going to be fatty acids. It's also made with a glycerol, so there's 3 fatty acid tails and 1 glycerol. The polymer are going to be lipids. They're going to be long chains of fatty acids. This is used for long-term energy storage. It's also used as protective coverings, like on the cuticle of a plant. They also form things like waxes. Steroids are considered part of this group. Those are important for hormone functionality. So examples would be butter, which would be a saturated fat, and olive oil, which is an unsaturated fat. So that's going to exist as a liquid at room temperature. Proteins are made up of amino acids. If I string a bunch of amino acids together, I get a polypeptide chain. This has many, many functions. So, for example, enzymes are largely proteins. There's also transport proteins in the cell membrane that let things into and out of the cell. Think aquaporin or calcium protein pump. But enzymes, proteins, these all have many, many functions within the body. So next question. Describe a situation that would cause a cell to increase or swell and decrease or shrink. So here I've got my cell. It's got 6 little particles inside. And I'm going to take that cell and I'm going to drop it into this first beaker. You see that there's much more particles inside the cell than outside of the cell. So to make up for that concentration difference, assuming that this red particle can't enter or exit the cell. The only choice is for water to flow into the cell. This, of course, causes for the cell to swell. In this case, we call this a hypotonic solution. Because the solution is less concentrated than the cell, water has to flow into the cell to make up for that concentration difference. I'm going to take that same cell, and this time I'm going to place it into a beaker that has many more particles in it than inside of the cell. And again, since these particles can't move, the only option is for water to exit the cell. So water goes out of the cell, causing it to shrink. In this case, we say that the solution was hypertonic, which means it was more concentrated than inside the cell. So water had to exit the cell to try to make the concentrations equal. Question 19 asks us to compare and contrast passive and active transport in terms of concentration, energy requirements, and the direction of movement. So, in passive transport, particles move from high to low. Active transport moves from low to high, so against the concentration gradient. No energy is needed in passive transport, whereas to move things against their concentration gradient in active transport, we're going to require energy, usually in the form of cellular energy, the energy that the cell uses, which is ATP. In passive transport, the particles move randomly by collision from high to low. And this can be across a membrane or from one side of the room to the other part of the room, but it's by that random collision, the random bumping together of particles. In active transport, particles are intentionally moved against the concentration gradient. So think of pumping calcium across a cell membrane. I'm intentionally moving those particles from high to low or low to high against concentration gradient. So types of passive transport that we covered, diffusion is the simplest form. Osmosis is just a specific case of diffusion where the thing that diffuses is water and it goes across a cell membrane. Active transport, the three that we talked about were endocytosis, into the cell. Endo means in, cyto means cell. Exocytosis, exo means exit, so out of the cell. And a protein pump, such as a sodium-potassium protein pump or a calcium protein pump.
Your unit test will be next class. This video will be posted on YouTube at LaFell Labs. Watch it over and over again. You can find the link on SRHS Bio. Check the bottom of the Unit 2 page and study. This is critical.